Hello, fam and friends. I have one question for you. This is my peach. Did you steal it? How is everyone? Welcome to a vaccine Kiki, or shall we call it a COVID Kiki. Chad Muckles here. So happy to be with you, everyone. Here I sit in quarantine with as many questions as all of you have. I've got quite a show rundown here. We've got an amazing lineup today for you of experts and scientists and people actually working on the vaccine. And I really feel that we are here today out of a necessity for good information and definitive answers. I think that we've all been sitting on our couches for going on 11 months now, seeing news cycles full of information, some of it good, some of it bad, some of it just complete conspiracy theories. So today, we are going to give you good, hard answers geared toward our LGBTQ plus audience. And again, I hope everyone's well. I miss you all. We're going to go over some quick house rules. I know that they played these for you before in the video, but I want to just run it all home with you. We will have moderators on all chat platforms. If at any time you are becoming unruly or troll-like in the chat rooms, you will be banned. We are here to have constructive conversations, to help provide factual information, and to eventually educate our family and friends. And if you are a troll tonight, you will get a lobotomy, just like that. So you don't want to be a troll tonight, okay? By the way, Nurse Ratched in the house, just for all of our essential workers on the front line, all of our nurses, doctors. Yes, it's drag, but it truly is from my heart a tribute to you all tonight. All right, in the house tonight, as you see, we have got Jennifer. Give it up for Jennifer, our ASL interpreter. Yes, mama. You are looking fierce, girl. Loving you. Welcome to the fam tonight. All right, I want to remind you all to stick around for the closing remarks at the end of our Q&A tonight, the portion of our Q&A. We also want to let everyone know that if you don't see your question or if you didn't get your question read, we will repost them to vaccinekiki.com and get everything answered as well as we can. So I think with that, we're going to get going and introduce our panel of doctors and scientists and experts right now. So I'm going to bring on screen right now an amazing doctor, Susan Little. Susan Little, MD, is a professor of medicine in the Department of Medicine, Division of Infectious Diseases and Global Public Health at the University of California, San Diego, UCSD. Since joining the UCSD faculty in 1993, Dr. Little has developed one of the leading and most productive National Institute of Allergy and Infectious Diseases sponsored programs in the United States for the identification, enrollment, and treatment of individuals with acute and recent HIV-1 infection. She directed an NIH-funded project at UCSD to evaluate determinants of HIV transmission and now serves as principal investigator of the San Diego Primary Infection Resource Consortium, a program focused on research related to HIV prevention, transmission, and pathogenesis. She also serves as the principal investigator on the AstraZeneca and Janssen COVID-19 vaccine trials at UCSD. Please welcome Susan. How are you?
Great to be here. Thank you so much.
So good to see you. You're in San Diego?
Correct.
We are so lucky with the weather. We wouldn't want to be elsewhere. I was noticing that in 1993 is when you joined the faculty at UCSD, correct? Well,
Correct.
that was the year I started doing drag, Susan. So does that mean I have my master's in drag by now? By the way,
Absolutely.
Thank you. By the way, I'm Chad Michaels, and I am not a scientist, ladies and gentlemen. All right. All right, Susan, I'm going to bring on our next scientist right now. Please welcome Jason Diaz, Ph.D. He's Assistant Professor of Integrated Science and Business and Technology at Salle University. Excuse me, I'm going to take just a quick sip. I'm knitting a sweater in my mouth, you guys. Okay. At the Technology of Salle University, Jason earned his doctorate in cell and molecular biology with a focus on virology at the University of Pennsylvania in 2015. His graduate studies centered on the regulation of life cycles of the human papillomavirus and recently identified Merkel cell I'm going to get this right, polyomavirus. I think that's right, Jason. At LaSalle, he teaches a variety of courses that explore advances in biotechnology and the translation of basic science research and therapies used in the clinic. Welcome, Jason. How are you?
I'm doing great. So honored to be here with you.
Well, we're glad to have you. I'm going to ask you a quick question, and I just want you to nutshell it for everybody who's listening. What is a virologist?
It's just a scientist who really specializes on studying viruses, just like the coronavirus.
Perfect. That's what they need to hear. All right, we're going to be right back with you. But right now, we're going to bring on our last scientist. And this whole thing is really mostly and partly his brainchild with Lucas Volk. And we want to thank Lucas for everything he's done to arrange tonight. Lucas is an amazing voice for social justice. And a lot of this is all him tonight, ladies and gentlemen. But we want to welcome Martin Naradikian, Ph.D. of Biotech startup LA. Martin Naradikian, Ph.D., received his doctorate in immunology at the University of Pennsylvania and did his postdoctoral fellowship at the La Jolla Institute for Immunology. While at Penn, he interrogated the B cell, which are the producers of the antibody response in the context of aging, autoimmune disease, infection, and immunization, including the mRNA vaccine platform. During his postdoctoral studies at LGI, Martin focused on identifying and engineering immune specificities against common cancer mutations. He is currently helping develop the next generation of genetically modified cell therapies against solid tumors. Say hello to Martin. Hi, Martin.
Hello, everyone. Thank you for having me.
You look great. You know what, Lucas? I can hear everybody but Martin, and I don't know why, but that's okay. Because I want to say, while maybe Lucas is working on the sound, happy birthday, Martin. Yesterday was your birthday.
Yes, thank you.
You are a gift to all of us. I'm sure that everyone else can hear you. So what I'm going to do is ask you really quickly, the same kind of thing I asked Jason is, what, in a nutshell, is an immunologist?
An immunologist is someone who studies the immune system, very plainly said.
Okay. You quit talking, so I'm going to assume that you're done with your answer. Can everyone else hear? Just a test, can everyone else hear me? Okay. We're going to go ahead and start answering these questions, ladies and gentlemen. Now, Lucas and Martin have collected questions from many, many people. Lots of questions to go over tonight. Like we said, if you don't see or hear your question answered, check after the show. We are going to start with, this is basically in 5 sections, and we're going to go with a vaccine overview, and we're going to include everybody in this segment. So our first question is, what are we injecting into our bodies? And what is the actual vaccine, and how does it work? And we're going to go to Jason first on this.
We actually switched it around. I'm
going to punt it to Martin to give us some background of where vaccines come from, and then I'm going to jump into talking about the Moderna and Pfizer vaccine. So Martin, can you give us some context?
Will do, of course.
Thank you, Jason.
So firstly, I want to say that vaccination is really an ancient practice. Some artifacts say that it can goes as far back as 200 BCE. And it wasn't until really the 1700s that Western Europeans started noticing this and rediscovering it from Western Africa and the Middle East. So it's been something that's been with us as a species for quite a while. Furthermore, vaccines themselves are just something that can stimulate your immune system to be ready for a future infectious event. So historically, that was, in those ancient times, was giving a small, harmless version of the virus, meaning, they would snort a little bit of an infected person's scabs or something. Gross, I know, but we've come a long way since then. Modern day viruses, or vaccines rather, are a piece of that virus, a small piece of usually a protein that cannot cause infection. So, and what has happened recently, and Jason will go into the details like this, is actually just taking it one step further and making the new platform, the RNA platform, really quite exciting. So Jason, please take it away.
Great, thanks, Martin. And as Martin just said, in modern vaccines, we're really focusing on giving people just one piece of the virus. For COVID, we're going to focus on something called the spike protein. So if you imagine, I'm just going to borrow the ASL symbol for COVID, right? These things here, these are the spike proteins that are grabbing onto your cells. I guess it's this way, sorry. So that spike protein, which is grabbing onto your cell, that's what we're going to focus on for all of our vaccines. But what we're doing special with the Pfizer and Moderna vaccines, we're doing something very different. Instead of giving the actual protein of spike, we're actually giving your cells the genetic instructions for that spike protein. And that sounds kind of crazy and maybe even a little scary. So I'm going to take a little bit of time to explain what's going on here. So the technology we're using is called an mRNA vaccine. You all probably have heard, I hope, that genes are usually stored in something called DNA. Well, in our cells naturally take that DNA, and instead of copying the whole damn thing every time we want to make something, we just focus on the one gene that we care about. So we make a copy of that gene in the DNA. That copy is in the form of what we call mRNA. That mRNA copy is basically a blueprint. It's going to tell the cell how to make the protein that we need. In this case, that protein is going to be spike. So what we're doing in these new vaccines, specifically the Moderna and Pfizer vaccine, is we're giving the cells just the instructions just for that spike protein. So it's not going to harm us. It's just one protein that's just not really going to do anything, but it's going to act as a target dummy for our immune system. One of the cool things about mRNA is that naturally in our cells, RNA actually disappears pretty quickly. We usually make the blueprints, we use them, and then we get rid of the blueprints really quickly. Like we're talking within hours. So when we inject the RNA into people, that RNA is really only going to be, in our cells for maybe a couple of hours, just long enough to make a first batch of that spike protein, and then those instructions are going to be gone. I also want to really emphasize that there's no way for this RNA to really change your DNA. There's no way in our cells for that RNA to be turned into DNA and affect your DNA in any way. So we're not genetically modifying anyone or anything. All we're doing is providing a very short-lived set of instructions to your cells to make this viral protein. Now, the reason why we do that is your immune system is then going to see these weird spike proteins that you don't normally make and be like, what the hell is that? That looks wrong. So then we're going to start generating that initial immune response. And we'll talk a little bit later about maybe side effects, but that's probably why you're going to feel a little bit sick sometimes after you get vaccines. Your immune system is really kind of ramping up. But then you get, you're fine after that. And now you have that first layer of defense so that when COVID actually does infect you later, you already have a really robust set of defenses against that virus. Let me just make sure I got everything. Yeah, I think that should be it. Oh, I'm sorry. The last thing I just want to know is what's actually being injected in you. So we spent a lot of time talking about the RNA. That's the genetic instructions. That needs to actually get into your cell somehow. So we coat it with these fatty lipids, which let the RNA get into your cells. And that whole mixture of RNA and then the lipids around it is in basically salt water that's perfectly calibrated to match the inside of your body. So that's all that you're going to be injected with is RNA, which will only live for a couple of hours inside you, some lipids to help it get into your cells to get the process going, and salt water. So that's basically all the major components. And that's in a nutshell how these vaccines are working.
Okay, so this is kind of a sister question, and I think this is probably a question that a lot of LGBTQ plus individuals are asking right now. Why was the vaccine for COVID-19 made so quickly, but we still don't have a cure or a vaccine for HIV, AIDS, and cancer? And I will direct this to Martin, Jason, and Susan. Martin, I still can't hear you, so I'm gonna bump off Jason. Jason, go ahead and bump off Martin when he's done to let me know he's done. This is the only way I can do it. So go ahead, Martin.
That's totally fine. Okay, so I want to just provide a little bit of historical context again about vaccines and why this was made so quickly. So biomedicine, the fastest ever the fastest ever approved vaccine it took 4 years, and that was back in the 1960s. Needless to say, biomedicine has come quite a long way since then. We understand a lot more, and we have much more incredible tools at our disposal. Just to give you an example, the Human Genome Project took over a decade and $5 billion, several labs working around the clock to sequence one person's genome. Now, you spit in a tube, ship it through Amazon, goes to 23andMe, we can sequence your entire genome. So that level of sophistication and the ability to actually do that is one of the reasons why this was able to be done so quickly. Another important point is that RNA is actually extremely easy to make in the lab. Like, I'm tomorrow going into lab, I'm going to be making a bucket load of RNA. It's actually pretty fantastic, and it's as far as research purposes and for this, and the fact that it worked is in animal trials and now in human trials is just fantastic. So it's a very easy to make, very quickly to make, and that is why. Now, the question kind of that's asked is like, well, why don't we have a cure for HIV or cancer? Now, these are very different beasts. HIV is a very different type of virus. It's highly mutagenic, and it also attacks the immune system. It hobbles the immune response. And cancer, similarly to HIV, is highly mutagenic as well, and it's almost unique to each individual. And it's not a virus. This is cells becoming cancerous. So making something a one-size-fits-all cancer therapy for everyone is just next to impossible. So we're talking about really apples and oranges here. And I'm going to let Susan, I think, is next about explaining a little bit more about why HIV. Then Jason will finish off with a little bit more information about coronavirus.
Sure, thank you. So I think, as has already been explained a bit by Martin, one of the biggest differences is the rate at which these viruses mutate or change in the human body. So HIV changes very rapidly. So as an analogy, influenza. So we get a new flu vaccine every year because influenza changes rapidly. And so we need a new flu vaccine every year. So HIV is sort of in that same sort of rapidly changing, but even more rapidly. HIV changes rapidly within the same individual over time, over the course of 1 year. So when we give people drugs, that's why we don't use a single HIV drug. We use a cocktail, as we say. So when we first started with HIV therapies and we gave people, unfortunately, single drugs, they rapidly became resistant to those single drugs because HIV develops resistance. It mutates. So HIV, that's, I would say, the biggest hallmark difference is that rate of viral evolution and mutations. And we know that coronavirus, the family, and SARS-CoV-2, the COVID virus, mutate much more slowly. So they're very different in terms of developing vaccines that may work against something that's a slow-moving target versus something that's a very rapid-moving target. The second big challenge is that HIV integrates. It integrates into the host genome and lives there forever, with rare exception, and we all know them, that the occasional 2 people that have been cured from HIV through heroic transplants. HIV lives in the human DNA forever. And so developing a vaccine that will prevent integration of the HIV into the host DNA is something that we don't need to worry about coronavirus. So it doesn't, as was already explained by Jason, it doesn't integrate. So we're just talking about a virus that replicates and typically not for long periods of time. So we'll get a bit later into talking about replicaps in people who are more immunocompromised, where replication may persist for longer periods of time. But in the average healthier person, viral replication occurs for a period of several weeks and then people recover or don't. But that's the end of it. And then replication is over. So it's a very different illness, an illness that lasts 2 weeks compared to viral replication in the absence of therapy that lasts for decades. So they're completely different illnesses. And I think I'll turn it over at this point to Jason.
Great, thank you so much. So I'm going to circle back briefly to what Martin kind of dropped for me about the coronaviruses specifically. We have been studying coronaviruses for decades. This really took off in the early 2000s with the SARS outbreak, because SARS, right, was caused by coronavirus. And so in the almost 2 decades since the SARS outbreak, there's been a lot of interest in learning more about coronaviruses and also testing vaccines. People have been trying to create vaccines for SARS and then later MERS, and these efforts have really laid the foundation and kind of a almost like a blueprint or a game plan for how we might tackle this novel coronavirus that is emerging now. And so I really want to emphasize what Martin mentioned, which was that we were really blindsided by HIV. We knew a little bit about retroviruses, which is the family that HIV is in, but the amount of knowledge we know about coronaviruses, today, or we'll say at the beginning of the COVID pandemic, is leaps and bounds more advanced than what we knew at the start of HIV. And the other, I think, really important aspect for why we were able to move very quickly, specifically with the Moderna and Pfizer vaccine, is again the fact that it's an mRNA vaccine, as I already explained. The reason why the fact that it's mRNA is especially helpful in having a rapid rollout is all you need to know is the genetic sequence of the virus. You don't actually need to grab the virus from a sick patient, grow it up, figure out what cells it grows in. You don't need any of that. All you need to know is what is the genetic sequence, pop the sequence for spike out, and then create a vaccine out of it. And that's exactly what happened. We were really lucky and fortunate in that doctors and scientists in China published the sequences of the coronaviruses that they found as early as, I think it was mid-January of last year. We're basically, actually, I think tomorrow is the anniversary of when the first COVID vaccine gene sequence was published. And literally within days, the folks who developed the Moderna vaccine were able to select the sequence that they wanted and start creating that mRNA to start testing it in animals and cells and things like that. So the technology platform really allows for rapid kind of production of these new vaccines to test. And we already knew so much about the coronavirus to get started that we were able to really dive into things really quickly. And I think that's it for this question. Thanks, everyone.
Yeah. It's amazing how quickly y'all have gotten it together. And just back to the HIV virus, I think it must be so frustrating for people working on, a cure for HIV. It's like chasing smoke, essentially, with the way it works. So anyway, thank you for everything that you've done. All right, we're going to move on to section 2 right now, which all these questions are kind of naturally flowing into the next. This section is kind of living with HIV and the vaccine. And we're going to be talking to Susan and Jason in this little segment. So my first question in segment 2 is there evidence that PrEP or herpes meds, Truvada and Valtrex, can block the virus? Can PrEP, herpes meds, Truvada and Valtrex block the virus? Let's start with Susan.
Well, the easy answer, no. Early on, when COVID first came on the scene, there was frankly a desperation to look at old available drugs and try everything that had antiviral activity and look to see if any of these drugs might work. So that's why a lot of drugs that were active against herpes, against HIV, all of these things were tried. And there were a lot of what we call anecdotal studies. You all know the hydroxychloroquine fiasco. So a lot of drugs were repurposed and tried, but when sort of really held up in the context of a rigorous clinical trial, no, there is no good data to suggest that those drugs are effective. And another repurposed drug, remdesivir, which is one of the drugs that has been shown to work in hospitalized patients, that's another repurposed drug that was used for Ebola. So I think all of the drugs that we are now using are really repurposed drugs that have been used again with sort of, some rational explanation for why they may work. But no, there is no evidence that any of the HIV drugs, therapeutic drugs that are now being used to treat HIV or PrEP or herpes work.
And I'll just briefly say, I think Susan covered all that, all that I would want to say on that question. We'll continue to be testing things as we can, but I think at this point, there's no clinical data that really suggests that they work. And it's really those clinical studies that really look at a lot of patients in a lot of different contexts that we need to rely on to be able to make good informed choices for healthcare for people. And right now, just the data isn't there, unfortunately.
Yeah.
Yeah, I actually, speaking of just bad information, I just had someone who recently had COVID tell me to go ahead and take hydrochloroquine if I contracted the virus because they took it and it worked for them.
Yeah.
And, I myself have RA, rheumatoid arthritis, and I've been prescribed hydrochloroquine as part of my triple medication treatment, methotrexate, sulfasalate, and hydrochloroquine. I actually stopped taking it over the summer when Donald Trump told everybody to take it. And actually, since I've stopped taking it, I've stopped having heart palpitations, mild anxiety attacks, and it has not affected my RA flares. So I don't know. In some way, I benefited from this lack of good information because I'm off the hydrochloroquine just because Donnie said to. Anyway, all right. I'm going to move into the second question in this section, and that is, should individuals living with HIV be concerned about vaccine efficacy? Susan, did I say that right?
Yes. So the answer is not yes.
Okay.
Yes, I'll answer the question. So overall, vaccine efficacy for both Pfizer and Moderna is approximately 95%. The good news is that people living with HIV were included in both of those trials. The bad news is that they were included in relatively small numbers. So in terms of AspenOUT and answering the specific question, do people living with HIV have the same vaccine efficacy that people without HIV had? We don't know the answer yet. In fact, for many people who entered the clinical trials who had underlying conditions, the numbers are yet too small. So and I would go on to say further, the people who were allowed to enter the clinical trials were only people living with HIV who had well-controlled viremia, who had undetectable viral loads and durably suppressed viral loads. So I think going forward, when both Pfizer and Moderna have emergency use authorization approvals. So as we go forward, we will get more data and more follow-up, and there may in fact be enough data in longer term follow-up to answer this question more accurately about specifically subgroups of people living with HIV, particularly if they pool the data across multiple clinical trials. But in the published Pfizer efficacy study, they didn't even analyze the data on people living with HIV. On the Moderna study, they simply pooled all the people who had underlying conditions, heart disease, lung disease, HIV, renal disease, things like that, and said that the vaccine efficacy was equivalent in people with underlying conditions as it was with people without, which is about 95%. So in general, the recommendation is that people living with HIV who have well-controlled viral loads should feel comfortable, should be encouraged to take these vaccines under the supervision of their healthcare provider. I think as long as they don't have any contraindications, which we'll get to in a bit later in the discussion, and any reasons that they shouldn't take the vaccine. I think there is a bigger question about people who don't have suppressed viremia, people who are truly much more immunocompromised, people who have ongoing viral replication and or very, very low CD4 T cells. And I think I may punt that a bit because that comes up and overlaps with one of our later questions about vaccine among people who are immunocompromised. And I'm going to say that most people living with HIV who are well controlled really have done, should be expected to do, I think, quite well on the vaccine, but we really don't have any specific efficacy data at this time.
Well, that still is promising for a lot of people to hear, so
Yeah.
that's a good thing. All right, we're going to roll. Thank you, Susan, for that. We're going to roll right into section 3 on vaccine effectiveness and duration. And we're going to direct these questions towards Susan and Martin at this point. So my first question is, how long is the vaccine expected to be good for? Susan, do you want to take that?
Sure. So I have a lot of questions that I don't know the answer to, so I'm sorry.
Yeah.
So at the present time, we don't know the answer to that. So the good news is the vaccines, Pfizer and Moderna, have both been approved, and the studies upon which they were based are still going on. So the people who are on those clinical trials will be followed, and we don't know how long the protective immunity will last. Now, I will back up and say what sort of how much antibody is protective. We don't know the answer to that either. Sort of what's the correlative protection? How much neutralizing antibody or how much antibody do you have to be protected? We don't even know the answer to that. But in the course of following these individuals, what one might expect would be that over a period of time, people who are protected by the vaccine, this 95% effective vaccine, will start to see more cases of COVID among vaccinated individuals. And presumably, that's when vaccine efficacy is waning, is to drift off. Mm-hmm. And we hope that'll be a long time from now. But honestly, we don't know how long protection will last. And lots of studies will be done looking at that exact question in participants of the original studies. And it's possible that 1 year, 2 years, somewhere in between, we may start to see that the vaccine efficacy is drifting down. And so rather than see a large number of people getting COVID again, the recommendation may be that people need a boost, need another vaccine. And so I think we may, unfortunately, need to anticipate the fact that these Moderna and Pfizer are both 2 vaccine scheduled series before you get protection. But in 1, 2, or 3 years, we may need a boost before, in order to ensure ongoing protection. Just a prediction. We have I have no idea at this time, but it's possible that protective immunity will wane, will go down over time, and we'll need another vaccine. But those studies are ongoing, and at this time, I don't think anyone has any idea how long protection will last.
I think everyone just needs to get into the headspace that we need to focus on now, get our vaccines done, and worry about 2 or 3 years down the line later, because
Absolutely.
You know where we're at, Susan. All right, this one is going over to Martin. Susan, I'm going to have you cue me when he's done answering because I still can't hear Martin. So Martin, my love, once you've had COVID, does this mean that you're immune?
Okay. So most likely, yes, but long-term studies are still needed. There's some amazing work coming out of some of my old institutes, like the La Jolla Institute for Immunology, University of Pennsylvania, even the Rockefeller University. They're publishing really phenomenal studies that demonstrate detectable immunity over in a cohort of patients that had mild COVID. And they're showing detectable immunity at multiple parameters at least 8 months post-infection. They're tracking individuals over time, and they're sort of drawing those graphs, and they're seeing that it's actually stable, and there's nothing to really suggest that it's waning. So in that regard, we think that indeed, once you do get COVID, you do have protection, at least, or immunity, at least for 8 months, and obviously we'll do the studies to track it for longer. Now, I know that there have been reports of reinfection, and I just want to say that these are exceedingly rare. And oftentimes the symptoms that the individual experiences are much less severe to probably non-existent. And I kind of want to just reiterate, like, we don't call it the novel coronavirus for nothing. We still don't know a lot about how this virus interacts with our not only our immune system, but our body and vice versa. So we're still learning a lot about it, but I think we have a lot of tools ready to understand it and get a really good idea of it.
And Martin's done.
Okay, I hope that now this is the second part to this question. I hope that you didn't answer it because I couldn't hear you, Martin, so forgive me if I repeat myself. But the next question is the virus when you have the virus, do you get the same immunity that you would get from having the vaccine?
Yes, so yes, they are different. So as you remember, Dr. Diaz was saying earlier, the vaccine encodes the spike protein. So when you receive the vaccine, you're going to generate an immune response against the spike protein. On the other hand, the virus is not just the spike protein. There's many other components to it. So when you get an infection, you will generate an immune response against not only the spike protein, but also all the other parts of the virus. Now, you're going to think, oh, that sounds better, right? Isn't an infection better? But there is no evidence that the vaccine can kill you or cause long-term problems, whereas the virus definitely can. So in the other aspect of this is that the vaccine is better because we know how much we're putting inside of you. There's no way for us to know at an infection event, like what your actual dose was and whether or not that is sufficient to generate a good immune response to protect you going forward. But we have a pretty good idea with the vaccine because we know what the dose is and we know what the response is. So that's all I have to say for that. I'll just do a thumbs up.
Okay, Martin, yeah. Thank you. I'm so bummed that I can't hear you. This is like awful. All I get to see is that beautiful face, all that hair, and just you're basically lip syncing for your life right now. With no, that's okay. All right, Susan, you're just the scientist I want to talk to right now. The next question is, does the vaccine stop you from being a spreader, or are you still a spreader with fewer symptoms?
Again, easy answer. We don't know. So unfortunately, the vaccine trials were set up with two endpoints. All of the studies, all of the vaccine trials have been set up to determine whether the vaccines reduce symptomatic COVID and severe COVID. They have been set up to look at asymptomatic infection or transmission. So one of the big concerns, I think early on, perhaps still, is whether or not the vaccines will decrease the frequency of symptomatic COVID, but in fact convert people, if you will, to being asymptomatically infected. So that in fact, they could end up being asymptomatic spreaders. Both Pfizer and Moderna are doing a little bit of work in this area to address part of this question, the asymptomatically infected question. So they're looking at these are randomized studies. People get vaccine versus SIBO, and they're looking to see the frequency of asymptomatic infection in both arms by looking at antibody at various intervals in the course of the study to see if people have become asymptomatically infected during the course of the study, including vaccine recipients. Now, if the answer to that is yes, people became asymptomatically infected, that still doesn't answer the question about whether they were transmitting during that period. If they were asymptomatically, it would be nice to know if they were shedding, if they were potentially transmitting. And to do that study, you would have had to have ideally been collecting either secretions, nasal or oral secretions, to measure the amount of virusations, which we weren't doing because they were asymptomatic, or B, measuring their contacts to know if they transmitted. And neither of those things were being done. So it would really require a different kind of study. And you could design this kind of study, but again, neither of the current studies is ideally designed in this way. So you could look at, for instance, the amount of virus, as I mentioned, in the nose or the mouth over a period of time, just randomly collected to see as a proxy whether people who were asymptomatically infected had higher levels of virus than people in the vaccine arm compared to the control arm. And you could also do a transmission study looking at household contacts, or people who lived in what we call sort of congregate settings, households, colleges, nursing homes, things like that. So those are all studies that could be done, and maybe they're being developed. I don't know yet, but they have not been done to date. So as of right now, probably the best way that we can estimate the question of asymptomatic spread is with mathematical models. And mathematical models, frankly, are often very, very good. So we can look at rates of COVID in the community based on penetration of vaccine in those communities, come up with some estimates of whether or not that sort of transmission is happening. And then finally, I'm not a big fan yet, but there are some thoughts that human challenge studies may need to be done to look at asymptomatic transmission. The reason human challenge, that is intentionally exposing someone to COVID and then looking at the response. Those are the kinds of studies that we do when we have a treatment that works, and that works 100% of the time. And we don't yet have great therapies for COVID. We now have great vaccines. We still don't have great therapies. Kind of the complete inverse of HIV. HIV, we have great therapies. We don't have vaccines. So I'm not a big fan of human challenge studies until we get to the stage where we can, with great assurance, tell someone that we're exposing that no matter how sick they get, we can treat, and they will not end up in serious critical illness. So that's kind of a long-winded way of answering your question, which is to say, we don't know. There are a lot of different ways of trying to get at that answer, but the most likely way we're going to get at it is looking at the rate of asymptomatic infection in the vaccine and control participants in the ongoing studies, and then using that data and using community data, doing some mathematical model studies.
Whatever information we don't know at this point, it's remarkable what we do know. And from a layman's perspective, listening to you, the amount of trials, the type of trials, and the conditions for all of those trials that you as scientists have to pursue is an amazing, it's astronomical, it seems, to collect information. So thank you to all of you for dedicating your life and all of your time to this, clearly. Susan, I'm just going to wrap this section up. Again, this section we've been talking about vaccine effectiveness and duration. This is, should I stay inside and quarantine after my first dose of vaccine, after the second dose as well? And then, should I still wear a mask, Susan?
Okay. So the word quarantine suggests the idea of infectiousness. And first and foremost, as has been already covered, there is nothing infectious about vaccines in any way, shape, or form. So quarantine is not the right word. No, you don't need to be quarantined. Yes, you need to wear a mask. So you will, with both Moderna and Pfizer, you will develop partial immunity within 2 weeks of your first vaccine. And then probably within 1 to 2 weeks, depending on which vaccine you receive, complete immunity within 1 to 2 weeks of your second vaccine. However, you do not get to take off your mask. We are going to be wearing masks, social distancing, and using regular hand washing for the foreseeable future, months and months to come. Why? For the reason that I just answered the last question. We don't know a lot about this virus. We don't know the frequency of asymptomatic spread. We don't know how, what kind of risks the people we are standing next to are. So if I take off my mask and I am an asymptomatic spreader and I'm standing next to somebody who's immunocompromised and more at risk of acquiring severe disease, there are so many unanswered questions that we don't know the answer to. What if the person next to me got vaccinated and didn't have a good immune response because they were immunocompromised? The questions just go on and on and on. So we all need to continue to wear our masks, need to continue to socially distance, and need to continue to use good hand hygiene. And that's going to continue for a very long time, despite the fact that we're going to be rolling out this vaccine for a long time. The estimation is that herd immunity, this idea that enough people in our community have been vaccinated that the virus cannot propagate, cannot spread within the community because people either have natural immunity or vaccine-induced immunity. Current estimates suggest that more than 70% of the population needs to be vaccinated or naturally immune. That's a huge number. And it's not 70%, average. There are probably pockets, there are, so in certain areas, it may be greater percentages, and in other areas, it may be lower. But just take it as a general, 70% or more need to be vaccinated. So until we hit that number, which is almost certainly 6 to 9 months away or more, we need to be wearing masks. It's just, honestly, it's irresponsible not to, because you don't know who you may be exposing and what their risks of infection may be, and the fact that they may not have had a good response to the vaccine, even if they've been vaccinated. So, so that's my long-winded answer to yes, you can't take your mask off.
Yeah, I'm here going crazy. I'm doing snaps. I'm banging the ceiling for you. But we can all, I think one thing that we can just, it's a yes, it's a for sure answer. If people wear their masks, what is the percentage when both people wear their masks? What is that percentage of protection? Like 97%, I think? 95? Am I wrong? If both people have a mask.
Yeah. If anybody else knows a number, I mean, it depends on the mask you're wearing.
Okay.
If you're wearing a really good mask, it's incredibly high. If both people wear their masks and stand socially distanced apart, it's incredibly good.
And it's not political, people. Do it for your mama. Do it for your mama. Yeah. Do it for your grandma. Do it for your friend's grandma. It just, come on. All right. Susan, thank you. That section was heavy on you, and you are amazing. Great. All right, we're going to move on to section 4, and we're going to be talking about COVID mutants and the vaccine. And we're going to be speaking with Jason and Martin. And we're going to have Jason take the lead and then cue off Martin. And then Jason's going to cue me when Martin's done. We know what we're doing here. All right.
I will definitely do that.
All right, Jason. So there's really just one question in this section, and that is, will these new mutations still be covered under the current vaccines?
Great question. And actually, before I launch into that, I was just thinking and kind of inspired by Susan's discussion just now about mask wearing. I think an analogy I want to just throw out there to the audience and my fellow LGBTQ community members and family, you know, with HIV specifically, we know that you need to get tested because we know we can spread that without symptoms. And even if you're on therapy and your viral load is very low, that doesn't mean that you're going out and having unprotected sex either, right? You're still taking precautions. You're telling people about your, you know, your particular situation. So just like we have already been grappling as a community with dealing with a virus that can be hidden and that we can spread to others unknowingly, and we need to take steps to both protect ourselves and others, that's exactly the same mentality we should be taking into the, you know, thinking about COVID, think about asymptomatic spread of COVID, and how we still need to be protecting both ourselves and others, even if we get vaccinated. So I just wanted to just throw that out there just to have you guys think about it. Let me return now to the question. Thank you. Then we return back to Chad's question about the mutants, because this is, of course, has a lot of people a little scared. I'm going to try and breeze through this because I want to make sure we have time to address the last questions about autoimmunity and allergies. Very simply, we have data right now to suggest that all of the mutants that we have seen will still be covered by the vaccines. How do we know this? Okay. First of all, we know that the viruses mutate. Dr. Little kind of mentioned this a little bit previously. The rate of mutation for COVID seems to be manageable. We seem to be able to keep up with it. It's actually remarkable that it really took a whole year for us to get, you know, a mutant that actually is starting to spread better, which I think everyone has heard of the variant that was identified in the UK. But really, the mutants that we only care about for the vaccines are the ones that happen with the spike protein. So again, remember this, right? So just the spike protein. And fortunately for the vaccines, we have samples of antibodies that humans have generated to the vaccine. So we're able to take actual antibodies that real people have generated to the vaccine and test them against new viral variants that come out. Now, there's always going to be a little bit of a lag time, and we're really testing those, you know, with cells in a dish, sometimes in animals. That takes a little bit longer. So the evidence is not always going to be 100% gold standard testing in a person, but it at least gives us pretty good confidence of what the situation is. And very simply, there are no variants, I think, out there that have been shown to escape from the vaccine, and we should feel pretty good that these vaccines are still going to be good for the rest of the year. The last point I'll make really quickly is the nice thing about the mRNA vaccines, again, is all you need, again, is that genetic sequence, which means that we can introduce a new vaccine with a slightly different genetic sequence very quickly if we do get a virus that mutates, you know, some type of protection. We already have a process sort of in place for flu. That process is pretty different from the mRNA process, but it's the same type of idea of, you know, we have the tools available to adjust our strategy in response to these new mutants. And so I'm going to turn it quickly to Martin for some last points, and then I'll turn it back to Jad.
Right. So you know how we've been talking a lot about the spike protein. Well, proteins, the building blocks of those guys are amino acids. And it just so happens that the amino acids of the spike protein is about 1,200. And these mutations affect about 9. These mutations in total affect only about 9 of those 1,200. You do the math, I did it for you. It's less than 1%. And the great thing about the immune response is that it is incredibly diverse. So all that rest of the sequence, all 99% of it, is open and the same. It's the exact same between the mutant variants and the vaccine. And your immune system is by design diverse, and therefore will most likely cover it. And I think data is coming out like that. But immunologically speaking, like it would be very difficult to imagine a scenario where this virus could really mutate completely away from this vaccine.
Thanks, Martin. We'll turn it back to Chad.
Very good. Okay, we're going to move right into, I wish I could hear you, Martin. Okay, we're going to move right into section 5, and this is talking about pre-existing conditions and the vaccine. We're going to be addressing Susan and Martin in this section. So my first question really, for me, totally hits home. So the question is, if I have an autoimmune disease, what concerns should I have about the vaccine? I personally, I shared with you, I've got rheumatoid arthritis. I know people with Crohn's disease. What say you, Susan?
You're
Susan and I actually kind of
going to hate this because Martin.
Well, here, how about this? How about Susan? So
Okay.
he can so Chad can actually hear you. Why don't you take this question, or that way, or do you want me to do it?
Oh no,
Okay.
I'm fine. Yeah. So the answer is sort of what I started in an earlier answer, which is to say, unfortunately, people with autoimmune diseases were excluded from the clinical trials. So to date, we do not have data on vaccine efficacy at all for people with autoimmune diseases.
Great.
However, I think at this stage, the recommendation really should be to talk to your healthcare provider and talk about the relative potential benefit of getting the vaccine and the likelihood of mounting an immune response. I mean, the negative probably is to not make an immune response. So we see this again in HIV, where we give vaccines. And the negative is that someone just doesn't make the protective immune response. So they don't get protected from hepatitis B or hepatitis A. And so, it's likely that we can, as appropriate antibodies, a serologic tests that measure antibody responses are made, that we may be able to monitor that. They're not going to be as yet as I don't know that we don't have the tests yet that can say, this is the level that you need to know that you're protected, but we at least could measure you did or did not make an anti-spike antibody. If you did not make an anti-spike antibody, you probably did not respond to the vaccine. So in that regard, that people with underlying autoimmune diseases could in fact be vaccinated and look into that sort of response. But again, I would advise talking, because autoimmune diseases have a wide variety from B cell to T cell, cell mediated versus humoral type immune deficiencies. And so I think talking to your healthcare provider about what the likelihood is that you will respond, and then trying again, proceeding and seeing if there's a way that you might be able to monitor the likelihood that you did develop a response. It's probably going to be difficult to know whether that response equates to protection, but if you make a response, an antibody response, at least that's something. Martin, do you want to add anything?
Yeah, very briefly. I think you already covered the main points, and essentially, it really just depends on the type of autoimmune disease that you have, and you should talk with your healthcare provider. And because the way they manifest and the treatments for them are vastly different, and they can affect your immune system in various ways. Some actually just like completely ablate or get rid of an entire cell type that is critical for your autoimmunity. So that's sort of your therapy, and to keep you from killing yourself. So therefore, like, you can't make a vaccine without that cell type. Or there are other types of therapies that would be very patient specific. So again, talk to your healthcare provider. And in the interest of time, I know Chad can't hear me. The last question. Let's go.
Okay, Martin, thank you. yeah, I'm definitely going to have to see my rheumatologist about this, but I can't see myself not taking this vaccine because, you know, I'm so chock full of medication at this point right now. One more vaccination can't hurt or kill me. And yeah, so we'll see what they say, but I'm confident. And, you know, I've been to see my rheumatologist lately and they haven't mentioned anything, you know, that it might be a detriment to me. So I'm keeping my fingers crossed and thanking you all for all the work that you've done. All right, Susan, we just have one more question and then we are pretty much done. And I'm going to tag a little end onto this one for you, Susan. Okay, so if I have severe allergies, should I be worried about taking the vaccine? Also, if I have cosmetic facial fillers, should I be worried about taking the vaccine? Asking for a friend.
All right. So I'm gonna say I'm gonna turf the facial fillers to who offered to take this. Martin, did you? Yeah. Okay. So, but the first, I'm gonna say that I'm gonna launch into a response about serious life-threatening allergies. But before I go there, the Moderna and Pfizer vaccines are incredibly safe. So these are really, really safe vaccines. They have a range of side effects which are greater than your average vaccine. So I don't want people to be put off by some of the stories you hear. Yes, there are fever, muscle aches, body aches, chills, headache, etc. But 1 to 3 days in general, self-limited, very safe vaccine. Now we move into the less safe part. Within the first 10 days of approval of the emergency use authorization of the Pfizer vaccine, there were 21 life-threatening allergic reactions, which in the medical field we call anaphylaxis. So that's when somebody's airway gets tight and without medical attention, they are likely to die. So these are serious life-threatening allergic reactions. The 71% of these occurred within the first 15 minutes. So these are immediate life-threatening reactions. That has led obviously to a lot of concerns about what kinds of allergies, people with what kinds of allergies should be concerned. First of all, the great news is that there is a reporting system that is collecting all of these allergic reactions so that we do get a good sense of how many there are, how bad they are. Pfizer is overrepresented because it was the first one approved. I'm aware of at least one anaphylactic or life-threatening allergic reaction to the Moderna vaccine. But there have been fewer reported because Moderna's been out for less time. So who should be concerned? And I'm going to read this because I think this is important enough that I want people to get it absolutely right. The CDC now warns that if you have these types of allergies, you should not receive either the Pfizer or the Moderna vaccine. If you have had a serious allergic reaction, anaphylaxis, or an immediate allergic reaction that is within the first 4 hours of receiving a vaccine, even if it was not severe, that immediate allergic reaction, to any ingredient in the mRNA COVID-19 vaccine, you should not get a COVID-19 vaccine. So as was explained earlier, so this is if you've had a reaction to the vaccine or an ingredient in the vaccine, and the vaccines contain genetic template, lipids, cholesterol, salt and sugar, and PEG, which I'm going to get polyethylene glycol, which I'll get to in a minute. And the polyethylene glycol is what most people think may be the source of a lot of these allergic reactions. Number two, if you've had a severe allergic reaction, anaphylaxis, or an immediate allergic reaction, even if it was not severe, after getting your first dose, don't get another one, okay? So if you had an allergic reaction after the first dose, don't get a second one. So the immediate allergic reaction within 4 hours is symptoms such as hives, swelling, wheezing, that sort of thing. the polyethylene glycol or polysorbate. These are what do you want to call them? They're something that is contained within a lot of vaccines and to stabilize. And so many people who have had allergic reactions to other vaccines may in fact have an allergic reaction to these vaccines. So this is something, again, if you've had an allergic reaction to a previous vaccine, it's really important that your doctor, and you can, or they should, look up what's in that vaccine, exactly what the ingredient list is. The ingredient list for these mRNA vaccines are public. You can look them up and find out exactly what's in them. So polyethylene glycol and polysorbate are the two compounds that are most likely the source of a lot of these allergic reactions. If you've had an immediate allergic reaction to a different vaccine or an injectable that's not related to COVID and does not contain polyethylene glycol or polysorbate, this does not mean that you can't get the mRNA vaccine. It just means that you should do so with some caution. You should talk to your doctor, your healthcare provider, and get some counseling about what the relative risk of an unknown risk of a reaction versus the possible benefit of this vaccine. The entire country is in the middle of a COVID surge, so the relative benefit of the vaccine is pretty high against a relative unknown risk of an allergic reaction. But in general, if you fall into one of these categories, again, you should talk to somebody about your potential risk. But you know, those are really the categories that I wanted to mention. And if you are worried at all, talk to somebody. And if you're going to get a vaccine, do it in an area where medical supervision is available. And you don't want to do it in one of the drive-through settings. You want to do it in a place where you can be properly supervised and medical supervision for, you know, emergency resuscitation kind of thing is available. So preferably a hospital-based setting.
Make a date of it. Basically, set aside some time. You need to stay the extra time after you get your vaccination to see if something happens. Okay. So I see Martin on my screen. Are you gonna tell my friend that I'm asking for if the vaccine is going to affect facial fillers?
Yes. Good news, everyone. You can be beautiful and vaccinated. Basically, the data on this it's just about 3 people on the Moderna trial out of 30,000, right? Reportedly had facial swelling. They had some had fillers before the vaccine, some had fillers after the vaccine. It's not really certain.
I want to hear. I can't hear.
And basically, the swelling and that they experienced was temporary and was similar to swelling seen with other vaccines. So these are people who had swelling in their fillers prior, and right. But that's the beauty about trials; they're transparent. They have to jot down everything, and so this was picked up. I first saw my alert on the local LA station, so that was funny. But yeah, I think we're going to be okay with facial fillers and the vaccine, everyone.
I wish I could have heard what you said. That's really the answer that I really wanted to hear. I tried to cop a feel off Adam's phone, and then his phone quit working. I'm going to watch the replay to get my answer. But thank you, Martin. You guys are amazing. Thank you for your time. I don't know if we have any other questions. Let me just get with Lucas really quick to see if we have any additional questions from our viewers watching this vaccination Kiki, where we donned our high heels, turned on the smoke machine, and locked the door tight. There's no more questions. All right, so yes. Okay, so I want to thank Jason, Susan, Martin for your time, your knowledge. I feel so much more informed, and I think that you guys have dispelled a lot of just straight up legends and myths that are floating around right now. So thank you so much to all of you. Really appreciate it. Yeah, and thank you to Jennifer, our ASL. You're amazing. Okay, so we have a little segment right now that I'm very excited about. So if you liked the show tonight, I know we all did the best we could. This is our first time, a little bit off the cuff, but if you did like it and you want to show some love, go ahead to our website and purchase a T-shirt. They are $10, and $10 of every sale goes to the UWC, the Unique Women's Coalition in Los Angeles, which is a trans organization led by trans women of color for the past 20 years. Right now, we have a very special guest with us. Please welcome Chanel Lumiere to tell us more about the organization.
Thank you all so much. My extreme gratitude for all the experts, Chad, Lucas, and everyone for putting this together tonight. Again, my name is Chanel Lumiere, and I am from the Unique Women's Coalition, UWC, founded in 1997. I think that makes us 24 years in existence. A little bit of our mission. Our mission is to elevate the voices and narratives of Black trans culture. And we do that through mentorship, scholarship, and community care engagement work. So events like tonight, this conversation like tonight is so important to our community. And donations made, T-shirts bought, help us continue the work that we do. So I'm, again, grateful to be in this space, and I thank you all for your time today.
Okay, thank you, Chanel. Chanel, unfortunately, I'm having a sound problem with you too. I couldn't hear you either. I feel so deprived over here, Mama. Thank you for being here. And Chanel, thank you for all that you're doing. We love you. We love our transgender siblings so very much. And just thank you from the bottom of my heart. We love you so, so much. All right, you guys, before we go, we have one final video. And this is a very special video for Martin. Yesterday was Martin's birthday, and I ain't gonna tell you how old he is because I can keep a confidence. But right now, right now, we have a very, very special video for you, Martin, from, dare I say, your biggest mentor in the world. Go ahead and take it away.
My God, it's like a bad Monty Python skit. Nobody expects their old thesis advisor. But here I am to, first of all, give a shout out to you and your other participants and organizers for the vaccine Kiki. I think that's a terrific thing you're doing. But equally important, I wanted to wish you a happy birthday. And so happy birthday, Martin. And to let you know that I'm looking forward, once we all get our vaccines and can travel again, to catching up over a good glass of wine. See you soon.
Amazing. Martin, happy birthday. Of course,
Thank
you
you
know,
for
your better half, the one and only Lucas, hooked that up for you. Are you crying?
A little bit.
Is that a tear?
Yeah. Thank you.
It's beautiful. All right, guys. Thank you so much for watching. I see that we peaked out. I think we had at one point almost 100 viewers. So thank you so much for watching. I hope that you feel more confident and more informed about the vaccine and COVID-19. I want to encourage everybody to wear your mask. Remember, it is not political. It is not political. It is for everyone's safety, and we want to keep everyone safe so that we can come through this on the other end in a new and better world. All my love to everybody. Thank you to all the scientists for being here tonight. Thank you to Lucas Volk for putting so much blood, sweat, and tears into this forum tonight. We appreciate you, and thank you all.
Thank you.
Thank you,
Thank
everyone.
you. Stay safe, everyone.